Ora si registrerà la conferenza.
So my name is Carlo Colombo and I'm the area manager for Prodotti Gianni. And I met Giorgio Virnicchi a few months ago. He explained me something I didn't know about Zebrafish model and all the applications that can be, all the improvement that can be achieved by using these animal models. I was amazed, so we decided to give this presentation together. Dr. Giorgio Virnicchi is very skilled about this model, and he has been working for several years as a researcher both in Italy and abroad. So I don't want to stay longer on this topic. So just, Giorgio, you can move on with your presentation.
Okay, first of all, thank you for inviting me, Carlo, and organize this nice webinar. So my name is Giorgio Virnicchi and I work for Plaisant SRL. Today I will give you a small introduction about Zebrafish animal model. In particular, I will tell you something about maintenance and development and spend a little bit more time on possible application. Before to do that, I just would like very briefly introduce the company from that I represent. Plaisant SRL is located in Castel Romano, nearby Rome, where actually we have a huge animal house made of 1,000 meters square, divided in 3 different areas: conventional area, specific pathogen-free area, and zebrafish area, here in blue. And what we do usually is support the researcher with different activity, from the more basic one, like breeding and management of animal colonies. We can perform health monitoring on your animal according to FELASA guideline. And all the administration of our animal colony is under a software called Mausoleum that usually we share with the client, so that they can actually see in real time our activity in our animal house, and also put some task directly to ask us to perform some task. Moreover, we also perform some chemical clinical analysis, and so we can provide organ and metabolic profile for you, different drugs treatment, blood sampling. We can do it weekly or at the time of sacrification of the animal, and organ sampling, of course, ex vivo. Then we have a nice facility for the generation of transgenic mice, where actually we can inject the zygote with different compound and change the DNA according to your request. And of course, we also perform embryo, sperm freezing, embryo transfer as well, and IVF. The majority of our work is on human tumor cell line. And for this reason, we have 2 xenograph model, the NSG mice and the sheet mice. And particularly the human cells that we use are usually transfected with luciferase, in order that we can evaluate the engraftment, the growth, and the metastatic process within our xenograph mice by IVIS acquisition. And then, of course, we can perform ex vivo analysis of the tumor. This is a lung tumor cell that is emitting the light, so we can detect them within the mice. And of course, then we can explant the organ and make different process for you. But coming back to the topic of today, just a small introduction about where zebrafish actually is coming from. Zebrafish is naive from the slow stream of the Ganga Basin in India. Different kind of zebrafish was identified over the time, but the fish actually became quite popular in science once George Sessinger decided to start to work with it in the 1970s at the University of Oregon. The reason why I'm telling you this is just for let you know that we are talking about an animal model that is very nice characterized, because people are working on it for more than 15 years. And just for make you a very brief summary of what was done, what was done in this time regarding the genetic manipulation of the model, and so by consequence the study of the gene function, until the time which in 2013 the entire genome of zebrafish was sequenced by whole sequencing, exome sequencing, and actually the author of this paper was rewarded with the publication on Nature. But more very nice insight is coming out from this work. Of course, there is some general information that the zebrafish sequence is large 1.575 gigabase, and within this quite compact genome there is more than 26 protein coding gene spread around over 25 chromosomes. But the more interesting information for us is that more than 70% of human gene have at least one orthology in zebrafish. If you consider that there is 80% of mouse similarity, this is quite nice, considering that we are talking about a small animal and a small vertebrate. And moreover, if you are interested in chromatin structure and how it can influence gene expression, it's important for you to bear in mind that also gene position along chromosome are often conserved in the zebrafish. So there is several reasons actually why there is some advantage to using a zebrafish. From the more simple one is the easy maintenance. They are smaller inside and they like to live in a close group in a large number. If you remember the picture of our animal house, you can see how much zebrafish you can fit in so small space. Actually, we have the capability of 1,000. There is a high number of offspring, and this is quite important because there is a lot of biological material with which you can work. It's reproduced quite frequently, like 10 days, and it's spawning between 200 and 300 eggs for a couple. Of course, if you need, you can shorten this time, but it's not suggested to because you are introducing some stress to the animal. But if you are in some emergency situation, of course, you can do it. The external and rapid development, of course, allow that in vitro fertilization and genetic manipulation is quite easy. Actually, we will give a look to that today. And more important thing, it's allowing you to save the mother after the generation of the embryo. This is quite important, particularly if you are dealing with transgenic animal. The optical clarity of the embryonic and the larval stage, of course, allow you to study by non-invasive live imaging some process that happen in a dynamic way. And also we will give a look to that. The high similarity with human genome, I already talk about the 70%, but I just would like to highlight also that the 84% of human disease gene have at least one equivalent in zebrafish. That make possible that today we have more than 15,000 mutants that are able to reproduce human pathology with kind of fidelity. And of course, zebrafish have also the capability to regenerate different organs. We don't have fin, but we have a central nervous system, heart, pancreas, liver, and kidney. And so it makes a suitable model for regenerative medicine and of course for study regenerative process. In fact, it's widely used in cardiovascular, neurological, and metabolic disease. High throughput drug screening, this is of course it's possible because for the small size of the animal, actually you can keep larval stage in 384 well plate, so it's very, very compacted, and the drug can be easily administered to the fish by dissolving it in aquaculture or by feeding. And again, more important for us, but this is also important for you, is that the institution on animal care and use committed recognize the zebrafish like a live vertebrate before the 5-day post-fertilization. And we will see actually how many things happen before this development time and how many things actually you can do. And this larval stage is handled with onset of the weaning, so at 6-day post-fertilization. So we was talking about the maintenance of the zebrafish. I told you that it's quite easy. Let's just say generally what you need to do in order to housing in your lab. As I told you, they can be stored in the tank. Male and female can be kept together. Also because they are easily recognizable by the orange anal fin and the yellow dorsal fin of the female, and in particular the size of the ventral size of the women is much larger, and this is actually the bigger mark of discrimination, and this is because for the edge storing. Of course, because you are doing research, you need to keep the zebrafish in the less stressful condition. And so temperature, pH, and conductivity of the water have to be under control. And luckily, we have a system that can do this for us, and it's able to detect any fluctuation and equilibrate the condition. Moreover, it's provided an exchange of 10% of water by day, and it's filtering and cleaning the water by carbon filter and UV light. But there is some parameter that you need to check actually, in particular the ammonia and the chlorine are highly toxic for the zebrafish. And considering that the standalone represent a unique biological unit, you need to be very careful, because once the chlorine will be toxic for your fish, will be toxic for all your colony. So you can risk to lose everything in one day. Of course, you need also to feed them. Feeding is quite simple. There is some kind of dry food, which composition is different based on the developmental stage. So you need to change the kind of food from time to time. And of course, you can do like with us in our facility. You can provide some living food like Artemia. Here you can see 44-hour post fertilization and 40-hour larva Artemia. The important to have living fish, living animal, living food is there is two main reasons. First of all, you are introducing food diversity that is quite important for the fish. It is less inflammatory compared to the dry food. And more important thing is pushing your fish to hunting for the prey. And this is a quite nice activity. It's important for their health, especially when you have when you are storing your fish in a limited space, like it's happening in the facility. And the last more important thing is the breeding. Breeding in zebrafish is also quite easily to set. You can go for one-to-one meeting, or you can go for group meeting, depending on what you need. If you need 5,000 eggs, you can go on the example on the right. But if you want to work with more handling biological material, usually you for your experiment you use a small tank. You can go up to 3 to 3 in the ratio male and female. The more important thing is to follow a simple protocol. The male and female have to be kept separated overnight, because zebrafish like to mate at the dawn. As soon the light, they are light exposed, so the light switch on in your facility, or you are exposing the fish to the light, you need to remove the room divider. And if everything is correct and your fish are healthy, in 25 minutes you will have mass egg spawning, and you can collect them, because there is eggs collector here and the sieve that avoid the mother and the father to eating the eggs. And of course, you need to fit all this activity in a circadian clock. We use 14 hours of light and 10 of dark, because it's what it suggests basically on the majority of publication. So very fast, let's go to the development. We know that the development is external and it is fast, but actually let's see how much it's fast. So in dark gray here, you have our post-fertilization. So as you can see, from the time of spawning where you have the zygote, this is the bigger one is the yolk sac, and this is the 1 cell stage. You go under cleavage division and you reach 1000 of cell at 6 hours post-fertilization. So that the gastrulation start and it's finished in 10 extra hours, so at 16 hours. And again, organogenesis starts very early, and it's completely at 2 hours fertilization. Just let me remind what I told you about the 5 day. So at this point, when all the organs are complete, your animal is still not considered by law a living animal. And because of optical clarity, of course, we can give a closer look to the stage of the development, and we can see that in 1 hour, brain here, notochord, somite, neural tube, and eye are already start development. But optical clarity, it's mean also that I can show you by a nice video of 24-hour time lapse cell division. And so you can see how the cleavage is occurring, and then you will reach the blastoderm stage. Here we are around 4 hours post-fertilization. Soon gastrulation will start, and you can already imagine that you can coupling to this imaging system some fluorescence marker in case you you are interested in developmental biology, you can follow the cell fate of the cell by expressing fluorescence specific marker. Soon will be possible to distinguish the animal axis. So this is going to be the head. You can already see the tail, the eyes and the brain. The notochord is here on the back, the tail. This is the yolk sac that it's for now it's providing the energy for this process. Soon the heart will beat. We are still before 24 hours, and the fish will start to perform his first movement. And this is actually will help him to hatch from the egg and go forward in development. So a lot of things occurring in a very short time. For this reason, the first use that was done in zebrafish and is still performing is the drug screening. The reason I already anticipated a little bit is because they are small and they can fit together in 384 well plate. Moreover, if you are studying, if you are performing a small exposition to drugs, actually you can even keep two fish together, and so basically doubling this number that it's already huge. And of course, you can screen a lot of molecules at the same time on the larger biological sample and select the drug that is more suitable for you based on the effect and of less toxicity effect. And maybe then, only then, select that one to try this drug on higher animal. This is just an example how this group was studying teratogenic effect of the ethanol exposition. So you have here 3-hour post-fertilization embryo. The reason why you started 3 hours, because now you can easily distinguish the good embryo from the not good one. Because if you have like massive embryo production between 200 and 300 eggs, of course, not everything will survive. So you need to make a selection before to do some drug treatment. And then after only 4 hours, they was able to observe the morphology of some organ, and they actually focused their attention on the heart. And yeah, this is information that we, of course, now we already know, but during embryo development, you shouldn't expose yourself and, of course, your kids to ethanol exposition. But of course, this you can do for how many chemicals you want, and you can do also more. Like this is another example. Make some study on the behavior. In this case, of course, you cannot perform it on 384 well plate because the fish needs some space to swim around. But so here we have a 96 well plate that still is a huge amount of number of animals, and so you can treat with different chemical or drugs, and actually check if you have a camera in the system how this treatment is affecting the motility of the fish. And this is particularly interesting if you are studying neurological disorder or some disorder that is affecting the locomotion. Moreover, of course, you can also perform aquatic screening, drug screening on the adult fish. Again, if you consider that the standalone is a unique biological unit, you need to perform this on the static tanks. Again, this is quite convenient because you can test a lot of animals at the same time, but of course you are spending a little bit more drug compared to the larval screening. Now I will show you just how you can knock down, so how you can affect gene expression, or you can modify gene within zebrafish. So the procedure is quite easy for 3 simple reasons. First of all, the embryo is huge. It's really huge. You can actually see it by your eyes, but of course, if you want to perform microinjection, you need a stereo microscope, but not so high magnification. And you can use a mold and an agar plate in order to have a scaffold shape like that you can see here, in order to place your embryo consecutively one after another. And then in a very fast way, because the development is occurring very fast, you can inject them. Usually people is able to inject between 100 and 200 cells in half an hour, if you have, of course, a nice system. And of course, depending on what you need and what you are doing, you can perform a yolk microinjection that of course it's less challenging because the yolk is more resistant by mechanical force and it's bigger, so it's more easy to inject something there. So if you are knocking down your genes and you are dealing with a small molecule like morphorin or sRNA, you can easily perform this. You don't need to have some kind of expertise. It's quite easy to learn. But if you want to make transgene, so you are dealing with big molecule. For example, in my experience, we was injecting Cas9 as a protein because it was more efficient. Of course, you need to inject your material in the one cell stage, and of course, it's more challenging. Then, according to what you do, you can just you can just screen your transgene or your overexpression, if you have overexpression of green fluorescent protein or other fluorescent protein, just by confocal analysis. But of course, you need to confirm by genotyping the mutation that you introduce in the fish. And again, zebrafish is quite nice in this because you can cut the tail fin of an adult fish that will regenerate in several weeks, or if you are hurry, if you don't want to generate big number of animals before the 5 day, at actually 3 day post fertilization, you can microsurge the tail of the fish, and you have 2 days to process your PCR, your biological sample, do your PCR analysis before to decide actually if the fish have the right genotype to be introduced in your system. And again, this is saving you a lot of time and reduce the number of fish that you are introducing in your study. This is just an example of how you can study, for example, hematopoiesis in zebrafish using some mutant that was generated in a similar way that we discussed before. So just really generally, hematopoiesis work in 2 waves of differentiation in zebrafish. Again, everything happened quite early. So at 10 hours post-fertilization, you have the primitive wave of differentiation in which from the ventral mesoderma, erythroid and myeloid line are generating. Then later on, after 1 day and a half actually, the hematopoietic stem and progenitor cell will arise always from the ventral mesoderm. And from that point, the entire line comprise the lymphoid line are sustained by this cell, exactly like in human. Of course, there is some difference, but what I would like to stress is the differentiative intermediate and the cytokine that are regulating this differentiation are quite conserved. And this is just an example of what you can do using some specific transgenic line. Here you have a CD41 GFP line that is a healthy marker of erythrocyte. So what you can see in this video is how from the ventral mesoderm the cell will start to express GFP protein, so they will start to be completely differentiated in erythrocyte, and how actually they can enter in the circulation. As you can see the cell that is passing the endothelium, and then they start to take the speed because they are moved by the blood flow. Particularly, if you want to go forward, you can follow their accumulation in the thymus, like the author of this paper did. And if you have a software that can track the GFP signal or other kind of fluorescent signal, you can actually track the cell within the fish. Of course, this is just an example because I would like just show you a small list of all the GFP, but you of course have also the RFP and other fluorescence marker that you can mark all cell type. In this case, we are in the field of hematopoiesis, but you can do basically with whatever cell you prefer. And you can decide, for example, to visualize macrophage, and using this mutant here, you can always study differentiation. But for example, you can also study how actually macrophages are able to reach some site of infection, and then you can make drug treatment and see in live how this process is affected by some effect of drug, or also knocking down some specific genes that are important, for example, for macrophage motility. Again, the possibility is a lot. You can see by your eyes some dynamic process and not only take a picture of it. And again, depend on what you want, you can do many things. Another fish that is quite interesting, particularly for us, because it's expressing the luciferase, and as I told you before, we have an apparatus that can detect the expression of luciferase. Is the Mytholuk GFP fish. This fish actually is quite recent, was published in 2021, and basically luciferase expression and GFP expression was introduced by Talent, and their expression is under the control of cyclin B. This means that every cell that is entering in mitosis will shine in green and will produce luciferase. So as you can see here at 6 hours post-fertilization or at 24 hours, the entire embryo is, you have massive proliferation, so the entire embryo is shining in green. But further in the development, this GFP signal will become more localized to the developmental tissue, that in this case is the notochord. In yellow, there is the generating fin and the brain. And of course, for test the efficiency of this mutant, they perform a control experiment doing some antiproliferatory treatment. And actually, they see here you are looking at a pool of an embryo, how actually the luciferase activity can be prevented by blocking the mitosis in the sample. For this reason, this fish is quite interesting because you can apply in different fields. For example, you can cross with different oncogenic mutant, and then, for example, that whatever cell oncogenic cell line you are looking will shine in green or will produce will produce light while will be in mitosis, so you will be able to follow your tumor growth and distribution. You can screen and evaluate possible antiproliferative effect, very similar to what they do for confirm the model. You can check actually if some drug have some antiproliferative effect that maybe was still not described. And again, you can use the regeneration capability of the zebrafish to study organ toxicity, because if your drug treatment is affecting some organ, will be cell death, they're apoptosing, and larva and the fish will try to regenerate it. So you will have emission of fluorescence again and light in the organ that are damaged of your treatment. And again, because I was told you, we work a lot with tumor cell. Zebrafish result be an excellent model also for xenograft. In particular, you need to make a small discrimination. If you are doing xenograft on zebrafish that have less than 10 days post-fertilization from the adult one, I will explain you soon why. Just to let you understand, the procedure is quite similar with other animal model. You inject your tumor cell in the fish. Usually you can inject in the yolk, and then you can observe the capability of your cell to grow within the injection site, to diffuse in the circulation, or to form metastasis. Of course, here the fish is transparent, so you can directly see your cell by fluorescence expression of a marker. But the real advantage is that there are several. The first one is that the zebrafish lack of adaptive immune system. If you think about what I'm saying, I did not speak about any particular xenograph mutant that you need. And the reason why is because you can take whatever fish line you prefer. As soon as you are before 10 days of life, your cell, the fish will not respond to the transplantation tissue. So your tumor can grow. Moreover, the zebrafish are capable to survive to higher temperature. Considering that you are dealing with the human cell, of course, have something that is more close to our environment. It's better for study grow and other behavior of grow engraftment and infiltration of the tumor in the tissue. And moreover, tumor transplantation, of course, because for the smaller size of the animal require only 100 of a cell. And this is actually another very great feature of the zebrafish, especially for us that we work with sometimes with the primitive primary tumor cell line. These cells are coming directly from the patent, and you don't have so many, and actually you cannot expand them. And of course, have a less number of injected cells allow you to perform more experiment always in the number of in the term of number of animal. Moreover, within 2 days, the embryo complete all the organogenesis, brain, heart, liver, but also circulatory and other organs are completed to the 2nd day. So again, you are still in this immunodeficiency developmental stage. And cancer progression, as I already told you, can be simply assessed if your tumor cell line express some fluorescence. So let's see just 1 second in practice what all these nice features can be applied. The author of this paper, for example, was just studying circulating tumor cell, and they was transfected to be shine in M-cherry. And because they don't need a xenograph, they decide to use this mutant, in which actually endothelial cells are expressing GFP. So what you see here actually is blood vessel of the fish, and in red you can see tumor cell. And you can decide to focus on each cell because you can do live imaging, and you can do time lapse of it, and basically see how the cell is moving to the vessel, how it is attached to the membrane, and how it interacts with it. And usually when you are talking about zebrafish publication, because of optical transparency, there is always a nice video that the researchers like to show. In this video, for example, you can see how red cell, tumor cell, can move between blood vessel of the animal from one vessel to another. Again, this is something that it's very important process to study in dynamic, and again, see the effect of some drug and how this can affect the behavior and the capacity of the cell to move and to pass the endothelium, and so to invade the other organ. It's quite important. And again, this is just an example. You can think, for example, to study solid tumor and see, for example, how the angiogenesis is rearranged in the site of tumor. Or, for example, you can have, again, GFP macrophages and see how this cell is interacting with the tumor cell. Then, if you want to do a xenograph, use zebrafish like xenograph model on the adult fish, of course, in that case, you need to have a particular mutant. And this is actually another confirmation of how much fish and mice and so human are similar, because the xenograph mutant actually have the same mutation that have the more widely used xenograph mice, that is the NSG. So you have the depletion of both genes that are important for the immune system. And in this way, the fish is completely immunocompromised. And some data actually see that tumor cells are able to have a similar growth and therapy response in fish. Of course, here you are losing the optical clarity of the fish because pigmentation is starting, and so you are not able to detect your cell just under a microscope. But actually, you can avoid this limitation, and by crossing your xenograph model with the Casper mutant. In this mutant, actually, the melanophore and the iridophore protein are completely lack because their synthesis is compromised. And that it means that light not only cannot be absorbed, so can pass to the fish, but can also not be reflected to your microscope. That basically allow you to have the best view of what inside your fish. And again, if you couple with the GFP or RFP tumor cell expressing, you can follow it. Again, so if you cross these two fish, and of course you need to inbred it and have the both all for the mutation in homozygosity, then you have something like that is very useful for you, because it's a xenograph fish that is still transparent also in the later stage. So then you can inject the cell. Here is just an example of eyes and abdomen that are the more common site of cell transplantation, but of course you can go directly in the organ, like in the brain, if you prefer. This is just an example that I would like to bring you here. So how actually you can see tumor growth, detect tumor growth and engraftment in the adult stage. So you can implant your tumor cell, you can check the fish that have a nice engraftment, and then you can start, for example, like they do here, some pharmacological treatment. And they can see here, like this is both their inhibitor of the part complex that is quite important for tumor growth. Respectively are tamatazole and olaparib. So you can see that both drugs actually are able to reduce the GFP signal that is coming from tumor cell. And of course, you can always do histological analysis ex vivo and have some other data. But this way of observation is still more suitable, basically. So just for concluding, zebrafish represent a good animal model to work this for several reasons. Of course, it's a vertebrate with gene functionality and signaling pathway are highly conserved. Just for make you a small example, you can inject human macrophage in immunodepressed fish, and they actually will be able to react and be activated and go in the site of inflammation. That it means that all the inflammatory circuit is maintained. Then the rapid development, of course, allow you to study biological processes in a short time. And this is actually also an advantage, especially if you want to study teratogenic effect or so on. The optical transparency, I give you some kind of example how fluorescence microscopy coupled with the transparency of the sample can be powerful to understand the biological process in their dynamic. And not less important, zebrafish represent a lower brain development animal. And this thing actually it make it easy to be accepted, approved in kind of experimentation. Moreover, help you to have a big number of animals that are authorized in your research. In this way, you can increase the number of animals. This is very good for the power of your data, because you are increasing the power of the statistic. But also, for example, in case of drug treatment, you are observe a more heterogeneous population, and also this is a kind of an advantage. And at the same time, you are reducing the cost, because the animal is it's small, it's cheap to maintain, and also if you want to do some treatment, the amount of drug that you are investing, it's limitless compared to the mouse, for example. But of course, the aim of this presentation is to highlight how much zebrafish can be good and suitable for the research. But the aim is not to replace the animal mouse model at all, especially if you are working in clinical research and trial. You need to move the knowledge that you achieve to higher develop a more similar animal like a mouse. But of course, it can represent a nice shortcut to a faster understanding of scientific questions that maybe are still not solved. And also, for example, like again, in the drug screening, can help you to reduce the number of your candidates before that you actually are doing some more expensive research. So with this, I would like thank you for the attention. And again, I will be pleased to take any question if you have, or you can contact me later if you have something to ask. Thank you again.
Okay, thank you, Giorgio. Can you click on the presentation now? I have a couple of slides.
Oh, okay.
Okay, so as Giorgio just mentioned, so if you have any question, you can just write it down on the chat, and Giorgio will answer at the end of this. There are just 2 slides I want to show. So as I told at the beginning, so Prodotti Gianni is a reagent company, so we sell reagents, mainly antibodies, proteins, ELISA kits, and something like this. And we have lots of reagents completely dedicated to zebrafish. So if you have any need, please just ask. So can you click again, Giorgio? Okay, and this is a gift that we want to give to our customers. So it's called the Gene Recommender. I think it can be very useful also for people working on zebrafish. So it's a website. It uses artificial intelligence to predict the needs of the customer. So Giorgio, can you click? So it's a website, so www.generecommender.com. You can just write down the genes you are working on and the disease if you want. Please click, Giorgio. So the artificial intelligence goes on the web and look for all the scientific data available on the web. So PubMed, preprint, genomic database, transcript database, and after its elaborations, it suggests you which genes are the closest related to the genes you put as input. So it's just a suggestion. You may use it, you may not. And after the answer coming from the AI, please click. Giorgio?
Yeah.
Okay, you can also go on the link, on all the link where the artificial intelligence has studied, let's say. So I think it's useful. I think it's very easy to use, and for the Italian customer working for public institution like university and so on, it's totally free. So you just need to go there and register. And okay, someone has asked the website. Okay, www.generecommender.com. So if anybody, I've done with it. So just click, Giorgio. Okay, thank you. So this is our company, Prodotti Gianni. We thank you for attending this meeting. I have a question for Giorgio.
Yeah, I don't see the chat.
No, I have it. I have it.
Ah, okay, you have it. Okay, sorry.
I can just say it. So as Plaisant, are you ready to perform drug screening on Zebrafish?
Let's say that yes, it's quite recently actually that we get authorization for breeding a zebrafish. So, but yeah, we have all the equipment in order that we can start the equipment and knowledge to start. So yes, theoretically we are ready in a very short time.
Okay, I have another question coming from Raefa. I have a question regarding the possibility of injecting drugs into the zebrafish embryos, and if there is a specific needle or approach that is best for that.
Okay, no, of course you can inject in the embryo, always in the yolk, and then based on the property of your chemical, it can diffuse or not. Maybe you can use some carrier in case you know that your molecule is quite struggling to diffuse. But theoretically, yes, you can inject in the yolk, and if there is a best time to do, actually, again, it depends on the effect that you want to see. If you want to see an early effect, I will advise you to inject the zygote, the one cell stage. But if you want to see, for example, an effect that maybe it's lethal on the embryo development, but you want to observe organogenesis, of course, in that time it's better if you can inject, and actually it's not so complicated still because the yolk is still quite big. You can inject in the yolk of 1 day post-fertilization larvae. So actually where actually already the brain and other organs start development. So is it possible in any stage? Someone is more challenging than another, but about chemical, you don't need, of course, to inject in the cell. You can inject in the yolk. So yes, is it possible? Inject drug and also cell, of course.
Okay, thank you, Giorgio. If anybody has some other questions, in the meantime, I just say that this webinar has been registered, and we will send the link to all the attendants. If there are no more questions, I thank you all for being here with us. I thank you, Giorgio, for giving this excellent webinar. and keep in touch.
Okay.
Bye. Bye-bye.
Oh, bye-bye. Bye-bye. Thank you for your participation.
You, bye.